Hello everybody, welcome to the webinar on When Analyzer 2 meets Matlab, Unleashing the Full Potential of Data Analysis and Visualizations. My name is Yinfen and your presenter for today.
Hello everybody, also from the moderation team. We'll be handling any questions that you have to Yinfen's presentation in the background. My name is Bastian, I'll be your moderator today.
Hello, my name is Fernando, also from the moderation team.
All right, thank you, Fernando and Bastian. Okay, we will start the webinar now and let me turn off the camera. In this webinar, we would like to showcase a variable feature of Analyzer 2, the Matlab transformation. We will go through Analyzer's Matlab dialog in detail so that you will be familiar with the variable options and their functionalities. We will provide exemplary use cases that cover pre-processing, data analysis, and visualization purposes so that you are well informed about the possibilities of integrating additional methods or views to Analyzer. Lastly, by demonstrating how to implement those exemplary use cases using variable Matlab functions as well as custom-made scripts, hopefully you will be able to make use of the transformation by yourself without problems. Now, let's take a look at the contents for today's session. As a starting point, I will give an introduction to the Matlab transformation. This entails informing you about the prerequisites for using it. After that, I will walk you through the user interface page by page. Next, I will demonstrate how we can make use of the transformation for pre-processing, analysis, and visualization purposes. In each session, there will be 2 exemplary use cases. A brief background information on the methods will be given. After that, I will mention some alternative interfaces with Matlab, mainly about exporting and importing data. Then I will conclude the presentation and provide some important take-home messages. At the end of the session, there will be time for questions and answers. Without further ado, let's start the journey. Signal processing methods are emerging continuously and rapidly. Incorporation of those methods or new visualization options into Analyzer requires considerable development time. Analyzer cannot have all of them. However, Analyzer offers a flexible capability to expand its functionalities via macros or solutions, add-ins, and the Matlab transformation, which is the focus of this webinar. With this transformation, it is possible to add further data processing functions to your pipeline, transfer your data to Matlab at any stage of processing, and perform further analysis steps, then import the results back to Analyzer and continue analyzing your data. It is particularly useful if you intend to reproduce results using other methods which are not available in Analyzer. By the way, the transformation can be found via the Transformations tab in the group Others. Please be aware that the Matlab transformation works with time and frequency domain data, but not with time frequency domain data. When transferring the data back to Analyzer, dimensionality and domain specificity of the data must be preserved. In order to use the transformation, you must have a valid license for Matlab version 7.4 or higher installed on your computer. First and foremost, let me walk you through the dialog pages of the Matlab transformation. In total, there are 3 pages. This is the first dialog page. On the top left corner, we can select the type of calculation to be performed. Either calculate and store the data when history node is created, or do not calculate the data and make it available only until requested to do so. Besides that, we can also choose either to raise Matlab desktop application or simply enter the Matlab code into the text box. To start a complete Matlab environment manually, enter the command desktop in the text box. One important note on the option to raise the Matlab window: if it's selected, the data export will be terminated by this message window, which must be confirmed in order to return to Analyzer. This is not recommended if you want to save the step as a history template for automation purpose. Instead of entering the code manually, it is also possible to load a script file by clicking on Load button. If you select the Calculate Data on Request option, the dialog contains two text boxes for Matlab code. In the upper text box, you enter all the Matlab commands that you want to be executed when the node is created. Please note that if you select this option, for performance reasons, no EEG data is made available to you at this stage. The EEG data will only be transferred to Matlab until requested by subsequent transforms or views. In the lower text box, you enter the code for the processing of this data. The second page is about exporting data and markers. By default, the EEG data is exported in a simple Matlab matrix format with the variable name EEG data. If the exported data is continuous, the data points correspond to the row of the matrix, and the channels correspond to the columns. When exporting segmented data, the EEG data will be a three-dimensional matrix, where the third dimension represents the number of segments. The Matlab properties variable contains the properties of the EEG data. They can be accessed and make changes if desired. If the export markers box is checked, a variable called markers will be created in Matlab. These two options here are used to improve the performance of the transformation in terms of saving storage space as well as reducing the memory usage for data transfer. We recommend that you only use this function if large volumes of data are to be transferred or if problems with main memory are to be expected. By the way, if you are using 64-bit system and by selecting packed data during transfer, you will get an error message that says that this feature has been removed as it is only needed on a 32-bit system. Data can also be exported in EEG Lab format. This is achieved by enabling the EEG Lab export options. In this case, different variables will be handed over to the Matlab workspace. On the final dialog page, we can select whether all the channels or only a selection of channels are to be exported to Matlab. If you only want to export specific channels, select the Export Channel Selection option. You can also use the Keep Remaining Channels checkbox to specify whether the remaining channels are to be displayed in the resulting dataset. Okay, hopefully now you are familiar with the interface and well informed with the options available in the transformation. How about start showing you what we can do with the transformation in the pre-processing stage. Analyzer offers several segmentation schemes, such as create new segments based on the marker position and create new segments limited by start and end markers. But let's say if you have a data set that contains no markers and you want to segment the data set into a specific number of equal blocks. In this case, you can make use of the Matlab transformation to add markers to the data. This will enable you to segment the data into equal blocks in Analyzer. So what kind of scripts we would need to prepare for adding the markers shown in the previous slide? Let me show you the codes and some important work concepts. Assuming that we want to separate the dataset into 3 equal blocks. So what we need to do is to add markers at the desired positions in the time axis of the dataset. Since we are going to manipulate markers, in this case is to add markers into the dataset, I would like to introduce you a variable called new markers that has a similar structure to the markers variable. The newly created markers are stored in new markers and read by analyzer. The first part of the script is about defining 4 new markers to be placed in all channels of a dataset. This is indicated by the value -1. Now, it would be the right time to talk about the indexing concept in Analyzer and Matlab. In Matlab, position and channel indexes of all markers are based on 1, whereas indexes in Analyzer are internally based on 0, which is according to the C# convention. Therefore, when creating the new marker structure, the fields position and channel need to follow the internal indexing logic of Analyzer. As an example, if you want to place the markers only in the first channel of the dataset, you would need to enter 0 instead of 1. The second part in the script is to define the markers descriptions. Here I have specified the first marker as begin, the second marker as 1/3, and the third marker as 2/3, and the last marker as end. The third part is defining the type of markers. In this example, they are defined as stimulus type. The last part is where the real calculation of the position is done. First, the length of each block should be determined. Then the position of each marker is calculated based on the length. Notice that the first position is set to 0, which means the first data point in Matlab environment. Now let me switch to Analyzer for the demonstration purpose. So the dataset we are going to use for all the demonstrations today is this dataset called as oddball rest subject 1. Let me open the dataset and activate the raw data. First, let me briefly introduce the dataset. The first part of the data is acquired from a resting state paradigm where participants were asked to keep their eyes open and closed. And the second part of the dataset is recorded from an auditory oddball paradigm. An auditory oddball is characterized by frequently appearing standard tones and rarely occurring target tones, which are often called deviants. The elicited ERP component, that is P300, is stronger for deviants. In total, the data has 32 channels, including 4 EOG and 2 mastoids. It is recorded with a sampling frequency of 1000 Hz, and FCZ was used as online reference. By the way, just want to let you know that I have prepared most of the Matlab transformation notes in the demonstrations today to save a bit time. And to make the notes more visible, I have changed the Matlab transformation color to red color. You will see it later. Let me open the note that I have prepared, which is this note, Matlab transformation data preparation, add markers. And I will click edit parameters and reprocess to open the node. So you can see the method codes which shown in the previous slides is shown in the text box here. So I will click next. It is not necessary to export anything and no need to raise EEG lab. So I will continue by clicking next. All channels will be exported. So I will click finish to complete the transformation. Please note that it might take some time to transfer the data from Analyzer to Matlab, and the data transfer is represented by a progress bar as shown here. Similarly, re-importing data to Analyzer will also take some time and shown by the progress bar. Okay, now it's done. We can check if the markers are added successfully. This can be done by right-clicking on the node and select markers from the context menu. So it's observed that all the markers have been added. Begin, 1/3, 2/3, and end markers. So we can use these markers to segment the data using the option based on start and end markers in order to get 3 different blocks with equal size. With this, I will end the demonstration and switch back to slides. The next use case that I would like to share is about implementing the re-referencing electrode standardization technique, abbreviated as REST, in Analyzer. It is an offline re-referencing method to approximately transform average or any scope points to a reference point at infinity, which is far from all the possible neural sources and has a theoretically neutral potential. Analyzer doesn't have a dedicated module for performing the REST method, but thanks to the authors of this article, a plugin that integrates the REST method in our Analyzer software has been developed. Specifically, the plugin makes use of Matlab transformation in a history template. Please note, this plugin is not developed by Brain Products. Hence, we cannot take the responsibility for the behavior and final output data generated by the plugin, and we cannot provide support if further questions arise regarding the functionality of the plugin. In this demo, I will show you step-by-step procedure how you can integrate the plugin into your Analyzer pipeline. As the first step, you will need to download the BP Analyzer plugin from the given link in the previous slide. Unzip the compressed file to the C drive. Make sure all files are in the C directory. Before applying the REST technique, it is required that the data is first referenced to an average reference. Please note that the option include implicit reference into calculation of the new reference should be selected, and do not give a name to the new reference channel. So now we are ready to the demonstration. Since the REST computation will take some time to compute, so I have prepared a shortened version of the data, which has been downsampled to 128 Hz here. And I have performed also the new reference transformation with average reference. Let me activate the node. As mentioned, the REST method is applied via a history template. So let me go to history template and open the history template. And drag the REST refer to node on the new reference node. So as usual, a Matlab message box appears stating data is transferred to Matlab. and you will see a window pop up. First, click on File and load Electrode file and open new chain.txt file. And after that, click again on File, but this time click on Calculate Lead field. At this point, I would like to ask for your patience, as this will take about some time to calculate. Okay, while waiting for the computation to finish, let me briefly tell you what is happening in the background. This step is about calculating the lead field matrix from the 3000 cortical dipoles and the given electrode array for the canonical concentric three spheres head model. The calculation is carried out in Matlab using the developed Matlab function, and this step will only appear when you use the history template for the first time or when the number of the electrode has changed. But to get more details on the rest method, especially the mathematical formulations, please refer to the published papers mentioned in the previous slide. Also, as stated in the paper, rest method has been applied in EEG studies of brain functions and dysfunctions such as schizophrenia, consciousness, and depressive disorder. And it is increasingly acknowledged by ERP's community around the world. And many studies have actually adopted REST technique to get zero reference as the foundation of their findings. So the calculation is still ongoing, but it will finish soon. Okay, good. So the calculation has completed. I will close it. As you can see, the data is transferred back to analyzer, and a node named REST refer to is generated, and it contains the zero referenced EEG data. Now let me overlay the data. And I will scroll through the data, and you will observe the data has changed after applying the REST technique. So to automate the steps, you can create a history template that consists of these two steps, which are new reference and also the REST technique, and apply it to the other history files simultaneously. So with the steps I showed just now, the REST method can be easily added to your processing pipeline. Now let's go back to the slides. So that was about using Matlab transformation for pre-processing. Now we will move to the focus towards adding data analysis methods using the transformation. In the final steps of ERP data analysis, you will require to quantify the amplitudes or latencies of the ERP components. These values will then go into your statistical analysis. There are many approaches that can be used for quantifying ERP components, but let's talk about two latency measures. The first one is fractional peak latency. The fractional peak latency is used to determine the onset time of components. The method finds the peak between a specified time range and then moves backward in time until the voltage reaches some fraction of the peak amplitude. The latency of this point is then used as an estimate of onset latency. So this figure shows an example of the fractional peak latency method. In this example, the onset time is determined at the latency when the voltage reached 50% of the peak voltage. Analyzer doesn't have a transformation for performing such quantification, but we can add this method in Analyzer easily with the help of Matlab transformation. In fact, we have a press release article on this method. It is published on our website. Let me show you. So the Matlab codes for performing this measurement are also available for downloading, which is the end files here, which is the name as fractional peak. Okay, now let's take a look at the codes in the M file. The percentage is defined by the threshold variable. Basically, it can be any value from 1 to 99. I specify it as 50%. So what should be done to mark the fractional peak? First, we need to have a loop which checks for all markers. If a peak marker is found, the value of the corresponding data point is needed from the EEG data variable. If you recall, this variable is used by analyzer for storing the exported EEG data in Matlab matrix. The fractional value then can be calculated easily and can be used as threshold. Then a second loop that searches for the data point in the backward direction at which the threshold is reached. This is the point where the new marker has to be placed. During re-importing, analyzer will look for the variable named new markers. Its contents will be added to the existing marker list. Recall that it must have the same structure as the markers variable. Now let's see how it's implemented using the Matlab transformation. I will tidy up the workspace by closing all the windows. And let me expand the whole history trees of the dataset. As you can see, the data has been pre-processed. The difference wave between the deviance and standards has been computed. And the P300 component peaks have been detected by using the peak detection transformation. Now let me open the Matlab node that I have prepared, which is this one, by clicking on Edit Parameters and Reprocess. By the way, instead of manually enter the lines of the codes, actually I have used the load button to load the M file. So let me click Next. Please to make sure that the option Export Markers is enabled. If you want to delete the old markers after exporting, you can enable this option here. Others options are not relevant, so they are not selected. I will not reprocess the transformation, so I will click Cancel. But we can check the results together. Let me double click on the node. So let me select one channel, for example, CZ, where the P300 component is pronounced. To check for the position and time of the newly placed marker, which is P300_50, one way is to point to the marker and then right-click and select marker information. And from here, we can see that the marker is placed at 256 milliseconds. And by the way, the amplitudes and latencies of all the markers can be exported using the peak information export or peak export solution available in Analyzer. Now, let's talk about the other latency measure. I will switch back to my presentation slides. Let me close the message box here. So fractional area latency provides a measure of latency by finding the point that dividing the area under the curve into specific fractions. For example, if you want to find the midpoint of a component, you could identify the point that divides the area under the curve into two equal regions, as in the example below. This would be the 50% area latency measure, and it works extremely well on large components like the P3 wave or on components that have been isolated by means of difference wave. Unlike the previous demonstration on fractional peak latency method, in this demonstration, I would like to show you how we can find the fractional area latency by using the ERP toolbox. It is an open source Matlab package which is integrated with EEG-Lab. Particularly, I will use the graphical user interface of the toolbox to perform the measurement. So to make this possible, EEG-Lab and ERP-Lab must be installed on the computer, and the path must be registered with Matlab, so that you will be able to directly open the EEG-Lab when you start Matlab. You can find more installation instructions on their respective websites. After everything is set, we are ready to switch to Analyzer for the demonstration. So on the same peak detection node, I will call the Matlab transformation. And I will load the code that I have prepared, which I save in desktop. And this is the code. So basically, it's a very simple code here, which consists of two lines. So the first line asks the Matlab to save the exported EEG data in text format at the specified location on the PC, and the file name is P300 underscore difference and underscore wave. I will need to import this data into ERP-Lab later. So, and I would like to have the Matlab desktop to be open when I run the transformation, so I enter the command desktop here. Also, the option show Matlab window should be enabled. Now let me move to the next page. As usual, I will select export markers and then make sure other options are not enabled. And I'll click next. And I will export all channels and click finish to complete the transformation. So now the data will be transferred to Matlab from Analyzer. So once the data is sent to Matlab, a dialog that I have shown you in the introduction will appear. And please try to resist and do not click OK until you are sure you are ready to close Matlab and start the re-import phase. So now the Matlab desktop main window is started. Let me call EEG-Lab by typing EEG-Lab in the command window. And on the GUI, the graphical user interface, click on ERP-Lab from the menu and select Export and Import ERP. Import ERP from the text file. So if you want, you can clear the data that you have loaded before. So you can clear the list and then add the current ERP file, which I store on desktop, and name is P300 difference wave. And since the data is have been saved in the multiplex form, this option is selected, which represents the points, represent the rows and the electrode it's represented by columns. And make sure the sampling rate is correctly stated. And our one is 500 Hertz, which is correct. And I will click OK. So we are asked to give a name to the ERP data. So I will enter, for example, P300 difference wave. And click OK. So now we are ready to perform the fractional area latency measurement. To do this, once again, navigate to ERP Lab, and this time choose ERP measurement tool. And make sure the current data set is selected, and under measurement, it's fractional area latency. By default, 50% has been specified, and the calculation is based on rectified area. And the measurement window, it's from 200 to 400 milliseconds, and the result will be sent to the workspace. So I will click Run. So the results are stored in this ERP measures variable for 30 channels. So these results has the unit in milliseconds. So to bring them back to analyzer, these values have to be converted to data points. And besides that, we will need to replace the markers position and description by creating the new markers variable. I have prepared a simple script to perform these steps here. So the first line here is basically the conversion from time to data point. And the second part is used to replace the markers. So I will run the script. Okay, now it is done. I'm ready to go back to analyzer, and I can click on OK. So a Matlab node is generated. Let me rename it to a telling name. For example, fractional area latency. So it's observed that the area latency 50 markers have been added to all channels. So from this example, we can see that a new analysis method can be easily added to analyzer with the help of a Matlab-based toolbox. So now let's move on to the next section. Now we are going to see how we can create additional views in analyzer. Analyzer offers a variety of views to display your data with a single mouse click. However, it is not possible to include all types of visualizations. But the good news is we can create additional views via Matlab transformation using the view functions available in Matlab. For instance, to create connectivity matrix views of different frequency bands, which will be shown in the following demonstration. By the way, we have also published a press release article on this topic, which can be accessed via the attached link. Let me show you. So the first part of the article is about visualizing epoch grids. And the second part is about displaying or creating the connectivity matrix view. And you can download the M files here, connectivity matrix view.m. The code can be easily modified to match other frequency band definitions. In addition, I will show you how to create interactive two-dimensional as well as three-dimensional plots of EEG data across channels and data point with the calculate data on request option. With this option, the transform is always applied to the portion of the data that is currently displayed. But before going to the demonstration, let me provide you with some relevant background information. Many functional connectivity measures are based on frequency domain data. To enable us to perform such connectivity analysis, we would need first to transform the time domain's EEG signal into frequency domain prior to connectivity matrices computation. For example, this can be done in Analyzer by using the FFT and coherence transformations with the following recommended settings. Now let's switch to Analyzer. I will close all the windows. So let me reprocess the node that I have prepared for this demonstration, which is this one, Matlab transformation connectivity matrix visualization, and it is applied on the coherence node. As you can see, the command line has been entered in the text box. It is the function that is provided in the press release article that I have shown just now. In this example, 30 channels have been used to generate the connectivity matrix. The second input argument indicates the connectivity method that was used in the coherence transformation. In this example, method is called COH, meaning that the option magnitude squared coherent was used. And EEG data carries the coherent values for all channel pairs. One thing to keep in mind about the downloaded function file, to use it, you would need to copy it to a folder of your PC and add it to the Matlab search path. So I will click Next. Again, nothing shall be selected on the second page, and all channels shall be exported. And I will click Finish. So this will take some moments to generate the view. By the way, the connectivity measure that has been used in this example is the magnitude squared coherence. But there are several other connectivity measures, such as magnitude of cross spectrum and imaginary part of coherency. If you are interested to learn more about them, I would like to invite you to watch our dedicated webinar video on phase and connectivity analysis from our webinar channel. Okay, I think we now have the figure. This figure displays the connectivity matrices, the coherence between all channels, pairs for the 4 different frequency bands. Coherence for each channel pair is coded in the color range from 0 to 1. A copy of this figure is also saved in the export folder of the current workspace. Note that in this example, large coherence values illustrate the impact of volume conduction effects on the estimation of EEG connectivity in the sensor domain. With this, I will move forward to the next demonstration, which is also the last one. Let me close the figure. So the last demonstration is about creating interactive two-dimensional and three-dimensional plots. I have prepared this Matlab node on the segmentation deviant node here. Let me open by clicking on Reprocess. And I will adjust the size of the text box. Okay, so that you can see all the codes in the lower text box. Okay, basically, we are going to create two surface plots. So the first one and the second one of the segmented data by using the surf function. The rest of the lines are basically used to configure the plot properties, such as edge color, line styles, and others. By the way, you will also observe that the option Show Matlab window is grayed out as the Calculate Data on Request option is selected. Now let me click Next to continue. None of the options here are selected as they are not relevant in this case. As usual, all channels are selected, and I will click Finish. So the data is exported to Matlab, and the view will be generated soon. Okay, we have the plot. So as you can see in the left plot, the channels are arranged along the y-axis, and the time are shown along the x-axis, while the plot on the right shows the channels as curves, one behind the other. If you want, you can also rotate the plot using the rotate tool here. So these two plots show exactly the same data and the same plot, but in each case from a different point of view. And the current figure shows the first segment of the data. So how about the plots for the next segment? Let me resize the window and so that they are side by side. If I move to next segment by clicking on the forward arrow here at the navigation bar, you will observe the change of the plot. So let me click to next segment. And again. So I'm sure you observe the change. So due to the option Calculate Data on Request, the Matlab plot shows exactly the data that you are currently see in Analyzer. This interactive visualization of EEG data across all channels and data points can be useful in observing the data continuously from one segment to another and to spot outliers. So I will end the demonstration and back to my slide presentation. Let me close the figure here. So far, we have seen how to make use of the Matlab transformation in expanding analyzer capabilities. By transferring the data to Matlab, perform some operations on it, and then return to Analyzer. But now I want to briefly talk about the interaction between Analyzer and Matlab in general, mainly on exporting and importing data. Under certain circumstances, you might want to export the data from Analyzer and proceed further in Matlab-based toolboxes. For this, we offer a solution called Create Mat File that allows you to export data in .mat format. It is part of our Analyzer Solutions package, which can be downloaded from our website. For example, this exported .mat file can be imported directly into the LiDAR software, which is a Matlab-based software for the analysis of skin conductance data. In Matlab, you can import the data using the load function. It is also possible to export the data in Analyzer's native format by using generic data export in Analyzer. Then this data can be read in EEG Lab via EEG Lab BVA I/O plugin. In case you have data sets that are not in BrainVision native format, but they can be loaded in EEG Lab, you can convert the data into our native format and import them into Analyzer with the help of the same EEG Lab plugin. Alternatively, you can write a brain vision file using the Matlab function write BVA. Now it's time to conclude my presentation today. The important take-home messages are the following. Analyzer offers state-of-the-art signal processing methods and provides a flexible capability to expand its functionalities via macros or solution, add-ins, and the Matlab transformation. We have shown in the demonstrations it is very handy to add methods and visualizations in Analyzer, either by writing Matlab scripts or making use of existing Matlab functions as well as toolboxes. This transformation allows editing single data sets, and it can be applied in template mode or via drag and drop. There are prerequisites for using the Matlab transformation. Please note that it works with time and frequency domain data, but not with time frequency domain data. The data dimension and type should remain the same when re-importing back to Analyzer. Importantly, it requires a valid Matlab installation and license. And with this, I finish my presentation, and we now have some time for open questions. Let me check the questions. Okay, so the first question is does it work with the pre-compiled standalone EEG version? Thank you for the questions. I'm afraid the answer is not possible to use the pre-compiled standalone EEG Lab version with the Matlab transformation. As mentioned in the presentation, to use the Matlab transformation, a valid license is required from MathWorks. Simply installing a Matlab runtime environment does not allow you to run the Matlab transform. I hope I answer your questions. Okay, I will look for the next questions. So the second question is also very similar to the first one. Does the Matlab transform also work with Octave? So my answer is still no. So this the answers that I gave just now holds true also for Octave. So the next question is about I want to use the automatic pre-processing methods from EEG Lab. For example, ASR in my Analyzer pipeline. Can I do that? Well, yes, you can you can perform the ASR in EEG Lab after exporting the data from Analyzer, and then open the GUI. That means raise the EEG Lab option and perform the ASR, and then back to Analyzer. By the way, it is also possible to automate this pipeline, this process. In EEG Lab, there is an option to print out the EEG Lab history to an M file that potentially can be used as a history template, so that all the GUI steps that you have performed do not have to be redone for each subject. Okay, there is another question. Can I use other filter methods as the one implemented in Analyzer with this Matlab transformation? Thank you for the question. Yes. So, for example, if you want to reproduce the results of applying FIR filter on the data, since we do not offer that, but you can export the data through Matlab transformation to Matlab, and then use the function Matlab function to design a FR filter and then filter the data with a Matlab function called filter, and then return to an analyzer and continue to analyze your data. I hope this answer helps. Okay. Yes, the next question is about exporting data. So it sounds like this. How can I export amplitude and latencies from ERP components for statistical analysis in Matlab? As mentioned in the presentation just now, I mentioned that the amplitude and latencies of the markers can be exported using peak information export and also peak export solution. And then you can load this data into Matlab. If you mean that you want to export the data, the latency and amplitude from Matlab transformation, that is also possible, but there will be a little bit more work to do. So after you export the data, the markers to Matlab, through the Matlab transformation, you will have a markers variable. From there, you can have the data point of the marker, which means that you are able to extract the information on time. So you will need to do conversion from time, from data point to time. And then to get the amplitude of the data, you will need to extract the values from the EEG data variable. I hope this answered the question. Let me check whether there are further questions. Okay, so there is one question. Will using the REST-REF EHTP in a history node from automatic processing, will the Electrode file be loaded automatically, or does that part of the process require the file to be manually selected for each participant? Yeah, okay, so you would like to use this history template that consists of these two steps, which is the new reference and also the REST method in the history template, and automatically apply to all the history files. Yes, that is possible. So you will need first to apply one time, and then for the next, for applying to other history files, it will be automatically done. So I hope this answers your question. So I will pause now, receiving further questions. I hope we have covered most of your questions. If we were not able to address them all, please feel free to send your questions to our email account via support@brainproducts.com. I hope you enjoy my presentation and are tempted now to try out the Matlab transformation. Let me go back to the slides and provide you with some materials for further reading. If you want to get more information about the transformation, the first resource that you can refer to is our user manual, chapter 7.7.3. And please check out this very useful press release article that I have mentioned in my presentation. To learn more about ERP analysis, connectivity analysis, as well as how to apply Hilbert transform in Analyzer, you are invited to watch the following webinar videos from our webinar channel. Here are the literatures and books that you can refer regarding the mentioned analysis methods. With this, I would like to thank you for your attention. You will soon receive a follow-up email with a link to the PDF slides and a video recording of the webinar. Please do contact us at support@brainproducts.com if you have any more questions about this webinar or other topics around Analyzer. Thanks again for participating in this webinar. On behalf of the whole Brain Products family, I wish you all good luck with your data analysis and research projects. I will see you around in our next webinar. Bye-bye.